All right, perfect. Okay, so I think yeah, in order to be more efficient in this lecture, so probably I will not stop to answer your question during the lecture. So in case you have questions, please type those questions in the chat box. Lei is going to help me to answer some of your questions. So each time, once I finish all the lectures, planned lectures, I will, approaching the end of the lecture, I'm going to leave some time for you. If you have questions, so we can discuss at that time. All right, thank you. Let's continue. All right, so so the first lecture actually is focused on the single gene inheritance, which is also called the monohybrid. So monohybrid genetic inheritance pattern. All right, so here we talk about one gene. So the mono here means one, right? One gene, but we have two alleles. So last time we talked about this. So we have two alleles for one gene. And this gene is actually determining the shape of the seeds of the pea plant, right? So the big R is the dominant one, and the small r, the lowercase r, is the recessive one. All right. So we start from two parents. Those two parents are two lines. All right. Okay. Now we cross these two parents, and we can generate the first generation, which is the F1 progeny. And as you can see, we start from two parents with two different phenotypes, round shape and wrinkle shape. However, when we cross these two parents, we end up with a F1 population, progeny population, showing only one phenotype, which is the round shape. So what does that mean? Based on what we have learned yesterday, so this means like this actually indicates the relationship between these two different alleles, right? The R, big R, the capital R is the dominant one, and the lowercase r is the recessive one. The recessive phenotype will be hidden in some generations, but the dominant phenotype will not. So you're going to see the dominant phenotype showing up in every single generation. So this is one criteria we can use to tell the difference between two alleles. Which one is the dominant one? Which one is the recessive one? The same for the phenotypes. All right. Now magic happens. So when Mendel do the selfing cross for the F1 progeny to produce the second generation F2 progeny. So what happened here? So as you can see, the missing phenotype reappeared here in the second generation. And Mendel did this experiment using a large sample. So how large it is, as you can see, right? You can do the math. So it seems like he did an experiment including about like 7,000 or 8,000 of individual plants. And based on this large sample, interestingly, he observed that, okay, so in the second generation, there are two phenotypes, and the ratio of the individuals carrying these two phenotypes is roughly equal to 3:1. So yeah, please remember these magic numbers. So we're gonna keep using these magic numbers to help us to help us understand the genetics behind, and also can help us solve the problems, questions in your quiz and in the exams, right? Okay, so okay, yes, as we discussed the last time, so Mendel actually did a lot of experiments based on many useful phenotypes. So here is the list of 7 phenotypes here. All right, for all of them, he did an experiment based on a large sample, and for all of these 7 phenotypes, he consistently observed same ratio, 3:1. All right. How interesting it is, right? So why? Yeah, this is like this. So we're gonna look into this. So here I'm gonna introduce a very useful tool to help us understand the genetics behind this 3:1 ratio. So this beautiful cute square, we name this beautiful cute square as using a name, the Punnett square. Right. Okay, so how do we understand this square? All right, so this square, so the when you look at the y-axis, which is this side, so this represented the two possible gametes that one parent can produce. So we are talking about the monohybrid, right? So looking at the first generation, so all the individuals in F1 population carries the same genotypes, which is big R and a small r. So let's look at a monohybrid cross. So if we look at this parent here, this parent, the female parent, can produce two possible gametes, either the big R or the small r, with the same chance. All right, and this is the same for the second parent. Let's say a male parent here. So for this parent, so let's look at the x-axis here. So the male parents can also produce two types of gametes. One is the big R, the other one is the lowercase r, with the same chance, half and a half, right? Okay. Now let's see what would the progeny from this cross, what kind of like genotypes can a progeny have from this cross? So there are four possible outcomes here. 1, 2, 3, 4, and so if you look very carefully, you're gonna see this one is the same with this one, right? Because for this progeny, so this progeny get a small r, the lowercase r from the female parent, but I got a capital R allele from the male parents. But for this individual, for this possible outcomes, this progeny get a capital R allele from the female parent, but this progeny get a lower R allele from the male. So if you look at the ratio of the genotypes, so this will give you 1 to 2 to 1 ratio. I hope this is clear, right? However, yes, this is for the genotype, the genetic makeup. However, if we look at the phenotypes for these four outcomes, possible outcomes, so all these three showing the same phenotype, right? Because we have at least one big R there. So the big R, the capital R, is a dominant allele. It will give you a dominant phenotype. So these three possible outcomes will show the same dominant phenotype. Only this one will give you the recessive phenotype. I hope everyone agree with me. And so now, if we look at the ratio in phenotypes, this will give you 3:1 ratio. So yeah, okay. So these two ratios are very important. Again, they are very important. Please, yeah, read the notes on your printout, because when I say this is very important, yeah, it is very important. You will see some problem relevant to this kind of ratios in your quiz or in your future exams. All right. So let's look into the details about why. So this monohybrid cross can give you a ratio like this, right? Okay. So as I mentioned, we have a ratio of 1 to 2 to 1, right? This is the genotypic ratio from the monohybrid cross. And if you look at the phenotype ratio, this gives you the 3:1 ratio. And these two ratios are both related to a magic number here, 2. Why is that? Is there any like biological reasoning behind? Yes, because we're talking about diploid organisms. And for each gene, for each locus, we have 2 alleles, right? So that's why. So when we talk about a monohybrid cross, it will give you some ratios that are closely related to this magic number, 2 diploid organisms. All right. Okay, so I think Lei has already helped you yesterday go over the cell cycle and also the mitosis and meiosis and the comparison between mitosis and meiosis. So this, so now I don't want to spend time on this because this is easy for us, right, to understand this. The only thing I would like to give you the only thing I would like to mention this is to give you a very small, a brief summary about the comparison between the meiosis and mitosis. All right. So first, this is a very important concept. I hope everyone can pay attention. The ploidy. So what is the ploidy? Ploidy is defined as the number of sets of chromosomes in a single cell. All right. For example, when we talk about the gamete, everybody knows, right? Gamete only has 1 set of chromosomes. So for any gamete cell, the ploidy is equal to 1. And in this situation, we use a very special name here called the haploid. Hap means 1, haploid, right? Now, when we look at a zygotes or a regular diploid somatic cell, there are 2 sets of chromosomes. For example, in a human cell, regular human cell, we have 2 sets of chromosomes, right? So in this case, the ploidy equals to 2, and we call this kind of cells as diploid cells. All right. Okay. Now meiosis and mitosis. Okay. So the key point for the comparison between meiosis and mitosis is that, okay, the ploidy change, the change in ploidy. So that's why I just pointed out ploidy is so important. So in the meiosis, you're going to see a ploidy reduction. So why is that? So I don't have time to go through the process, but I hope everyone understands the process of the meiosis. So if you look at the diagram showing the meiosis, so you're going to see there are only 1 round of the DNA duplication. So pay attention. So during the meiosis process, there is only 1 round of the DNA duplication, but there are 2 rounds of the cell division. So this situation will lead to the ploidy reduction. So the ploidy in the meiosis process will be reduced by 1/2 from the 2n to 1n, right? However, this is different in the mitosis. Mitosis, if you look at the diagram, you're going to see there is only 1 round of the DNA duplication, and there's only 1 round of the cell division. So what's the consequence? What's the results from the mitosis? So we're going to see 1 cell become 2, right? A mother cell will become two daughter cells, which have the identical genetic makeup. And in each of the daughter cells, you still see that the 2 sets of chromosomes, 2n, diploid, right? So in the mitosis, the ploidy maintains. Okay, all right. So this is the only thing I would like to summarize for the comparison between mitosis and the meiosis. All right, so this slide gives you a list of concepts. So I don't think I have time to go over them one by one, but make sure after the class, make sure you can test yourself to see if you understand each of this. Those are very important concepts. So make sure you understand each of this, and this will help you to do well your quiz in your next in the next quiz, which is next week, and also will help you to solve problems in the exams. Right? Okay, so as I mentioned in lecture 1, we focus on single gene inheritance. So we are talking about just one gene, 2 alleles here. This is a very simple case. All right. We use the Punnett square to help us to understand what is going on behind. All right. So I believe Lei also go over some statistical rules with you yesterday. So here, I will very briefly to mention this rules, which this important statistical rules that can be applied in genetics, genetic analysis. Right. The most important rule in statistics is called the multiplication rule. All right. So in this rule, it says if we want to calculate the joint probability of event A and event B, so the P means the probability. So we want to calculate the joint probability that both A and B, 2 events, are occurring simultaneously. We use this notation to represent this joint probability. So if the event A and the event B are totally independent with each other, then the joint probability is easy. We just apply the multiplication rules to multiply the probability of event A by the probability of event B. So so if I just give you this, equation, probably it is very difficult to understand, to digest. So let me give you examples. This can help you understand this. All right, let's do a simple experiment here. Very simple. So if you have a coin, you toss it, and if this coin is a fair one, which means both sides have equally a chance to be faced up. All right. So if you toss this fair coin twice, so if I ask you what is the probability that you're going to get 2 straight heads, so I need you to calculate the probability for me. So in this case, we can apply this so-called multiplication rule. All right. So here we want to calculate the joint probability that the first toss end up with a head, and also the second toss also end up with a head. So this is how we can apply this rule here, right? But what is the logic behind? Oops. So we can use the square as well. This time here we can utilize this beautiful square in this situation as well. So if you look at the y-axis here, so this shows you the possible outcomes from the first toss. If this is a fair coin, so you have equal chance to get a head or tail, right? And the same for the second toss. And we believe the first toss is independent of the second toss, which means the outcome from the second toss does not depend on the outcome of the first toss. I hope you agree with this. So if this is the case, so we can see there are 4 possible outcomes, right? And the probability of this outcome, so this is the interested outcome we would like to calculate the probability for. And all right, so if you just look at this square, you can immediately tell me the number of the probability, because this is just 1 out of 4. So the probability to get this outcome, 2 straight heads, should be 1/4. So that's very straightforward. But this number can be calculated using this equation. Why? Because so for the first one, if you get a head, the chance of that is 1/2, right? For the second toss, if you want to get a head, the chance is also 1/2. If we believe these 2 tosses are independent from each other, then we can simply multiply these two numbers, and this will give you the same answer. Okay, so I hope, yeah, this is clear. And now we try to extend this example to our own business. So don't go too far. So let me pull you back to our business. So here we try to use this magic square, the Punnett square, to help us to understand what is going on behind this single gene inheritance. All right. So here, the first parent, let's say this is a female parent, and this is a male parent. All right. If you think about, so the female parents can give one of two alleles to the next generation, right? And if you think about this activity as a tossing coin, the same. So the first parent toss a coin, and there are two possible outcomes which have the same equal chance, 1/2 here and 1/2 here. And the same if you think about the male parent, the same. So you toss a coin here the second time. So two possible outcomes or two possible alleles here, and also have the same probability. Then you can use the statistical rule, the multiplication rule, to help you to calculate the probabilities. Right? It's easy, right? So if in the quiz or in the exam, if I give you some question like this, single gene inheritance, and if I ask you to calculate the probability that a progeny carries the genotype like this, it should be easy for you, right, to do the calculation. All right. So it is not hard, right? Okay. Now, if I ask you, because during the quiz or exam, so this is a challenge for us this quarter, because we have to handle everything online. So that means like I have to work hard to make like multiple versions of the test for you. So maybe in one test, I will give one version of the test. I will give you like, yeah, please calculate the probability of a progeny carrying this genotype. So you need to use this to calculate this number, right? So in another version, if I ask you to calculate the probability that any progeny carries this genotype, you need to use almost the same equation to calculate the probability for me, right? And in the third version, if I ask you to calculate the probability of heterozygous individuals, then using this multiplication is not enough, because in this Punnett square, we have two outcomes carrying the same genotypes. That means like you have to multiply this value by 2. So to get the 1/2, 1/2 of the probability to get the progeny carrying this genotype. All right. I hope this is clear. All right. So this slide, again, this slide gives you a list of concepts here, very important. Make sure you understand all of this, and many of this will show up in your coming quiz, and in the midterms. All right. So here, I just want to point it out, because in lecture 1, we focused on the one gene, the single gene inheritance, and we are talking about the monohybrid cross experiment. And in this experiment, you're going to see two important ratios here: 3:1 ratio, which is the phenotypic ratio. Why is that? So yeah, you need to understand this. And also, if we look into the genotypes, the genetic makeup, the alleles of the F2 progeny, so this gives you another ratio, right? 1:2:1 ratio. Yeah, those two ratios are very important. We're going to keep using these ratios in the future, and this can help us to do a lot of genetic diagnosis. So we're going to talk about this later. So yeah, trust me, those ratios are very important. Please make sure you memorize those and you understand what is going on. behind these two ratios. Okay, now, yeah.
Yeah, sorry. So anytime we have a 3-to-1 phenotypic ratio, we call it simple dominance.
Yes.
Right?
So if I understood, so you, I didn't hear you very clearly, but if I understood correctly, so you're talking about the 3-to-1 ratio, right?
Yes.
Okay. So here we so the 3-to-1 means like the two phenotypes. And one is dominant phenotype, and the other one is the recessive phenotype, right? So if we just look at the phenotype level, so this will give you the 3-to-1 ratio in the monohybrid cross. However, if we look into the genotype, genotypic ratio, so genotype is the genetic makeup, right? We're talking about two alleles combination, either the capital R, or the big R, small r. So if you look at this will give you same population, will gives you a different ratios here.
Right. My question was, so simple dominance, like the term when like you're going to refer to simple dominance, we should assume it's a 3-to-1 phenotypic ratio within a monohybrid cross. Correct?
Yes. Okay. Yeah. So yeah, this is a good question. So in reality, we can have very complicated ratios because there are too many kinds of traits, right? But here we just focus on very simple model. So here we talk about just one gene, two alleles, and a simple dominance and recessive relationships. Okay. So yeah. So in the future, when you continue a career in genetics, so probably you're gonna encounter a lot of like complicated ratios. So yeah, but still you can apply the simple rules to those complicated situations to help you understand the genetics behind. Right. Okay, so based on all these experiments, Mr. Mendel proposed his first law, the law of segregation. So let's see what does this law say. All right, so oops, where's my, it's here, oh, it's here. Okay. Let me relaunch this. All right. Okay, so let's look at the law of segregation, the first Mendel law. So this law says, okay, so this law actually describes the situation which is going on during the meiosis. So during the meiosis, so we start from 2N, right? The diploid, but we end up with 1N, the haploid. All right. So initially we have two alleles at one locus, one gene, two alleles. But during the meiosis, these two alleles, these two gene members, we segregate. And this kind of segregation is at random. So this is basically the idea that this law can tell us. So which means like during the meiosis, during the production of the gametes, the two copies, two alleles is gonna segregate randomly, right? So now we have two very important concepts here: so homozygous and heterozygous. So last time I remember some student asked me the difference between the homozygous, heterozygous, and homologous. So those are totally different levels of concepts, right? So when we talk about the different chromosomes, if two chromosomes are paired, they are similar to each other and carrying almost the same genes, we call those two chromosomes as the homologous chromosomes. However, if we compare two different chromosomes, they are non-homologous chromosomes. So we are using the homologous at the chromosome level. But here, when we talk about the homozygous and heterozygous, this is not the chromosome level. This is not the chromosome level. Sorry, this is within the chromosome. We're talking about a single locus where a single gene is situated. All right. for this single gene, we have 2 copies on 2 homologous chromosomes. So if these 2 copies, 2 alleles are identical, then we say, yeah, this loci is a homozygous loci. Otherwise, if the 2 alleles are not identical, so we say this loci or this gene is a heterozygous. And this, so please pay attention to this difference. They are very important. Don't be confused about this too. Okay, now, so yes, so in Mendel's experiment, we have round shape, we have the wrinkled shape, and we knew this shape is determined by a single gene. We have 2 alleles, the capital R and the small r, and the capital R will give you the the dominant phenotype, which is the round shape, and the homozygous little r can give you a recessive phenotype, which is wrinkled seeds. And interestingly, the wrinkled seeds are much more sweeter than the round shaped is. So yeah, we're going to talk about this a little bit later. So we're going to show some interesting biochemistry behind. So this can help you next time you go to the market store. So if you prefer some sweeter piece, so yeah, just so you know what type of the seeds you need to choose, right? Okay. All right. So okay, so different alleles. So when we talk about a gene, so the gene can take different forms of alleles. So why is that? So why a gene can, we knew like the alleles can, the different alleles account for the variability of the phenotypes, right? But why? How come a single gene can end up with different alleles? So yeah, let's look at this. All right. So usually we say an allele is a wild-type allele if this allele is very commonly seen in the nature, right? And usually, I mean usually, not always. Okay, the wild-type allele will be functional. So usually, in usual case, a wild-type allele is functional. It can produce some functional protein or enzyme, and this functional protein can do something in the cell to produce a kind of like a phenotype. So we call that phenotype as a wild-type phenotype. But wild-type phenotype is not necessary. It's not necessarily to be the dominant. Okay, so don't be confused about these 2 concepts. The allele, one allele could be wild type, but the wild type phenotype is not necessarily to be the dominant phenotype. Right. Okay, now, so if this gene and if the wild type allele has something changed, for example, the mutation, if a mutation occurs within this wild type allele, then we'll make another allele, right? And that allele is not wild type, is a mutant. So we call that kind of allele as mutant due to the mutation. There are many kinds of mutation. We're gonna learn those later. But yeah, mutant is caused by mutation. All right. In many cases, a mutation will have some effects on the function of this allele. So for example, if a mutation is fatal, so it can totally destroy the function of this allele. So this mutant allele cannot produce any functional protein. And then the cell probably will lose that function and will give you some other phenotypes. All right, that's the mutant phenotype. So this is how and why for one gene we can have different forms. We can have different alleles. Yeah, for most of the case, this is due to the mutation. All right, so what is mutant? So we need to learn the definition for the mutant. So this is what we can learn from the movies. The mutant are monsters. So if you read the newspapers, probably you can have some other ideas of what is mutant. Mutant is something showing abnormal phenotypes like this. All right. So if you ask the same question to the Dr. Google, the Google will give you some definitions, but sometimes the definition can be very vague. All right. But we can define mutant using the biological definition here. So this is the best definition that we can use for defining mutant. The mutant refers to any organism with a new or different characteristic because of mutation. I hope this is clear to you. All right. So let me give you an example. So the wild type and mutant is a relative terms. Sometimes, we might be wrong in terms of these two terms. So because the wild type is somehow like very arbitrary. So for example, if we are talking about rose, this is the rose that is most commonly seen in wild place in nature. So actually, the wild type rose look like this, and the beautiful roses you see in the market store are mutant, right? So sometimes if you go to the store and buy a bunch of beautiful roses for your significant one to show love, but you don't know they are not wild type, they are mutant, they are monsters. All right. So here I just want to let you know, okay, the wild type and the mutant are two relative terms, and sometimes we define the wild type very arbitrarily, right? Okay, so as I mentioned, so the mutation can cause some mutant alleles. So let's look at an example to understand the molecular basis for producing different alleles. So let's just use the round shape versus the wrinkled shape of the pea seeds. All right. So we use the same letters here. The capital R, actually in the pea plants, the capital R is a wild type allele, is a gene, is a wild type allele for one gene. And this gene actually, so this big R is a functional allele, wild type allele. It can encode an enzyme in the starch biosynthesis. So if an individual has this R allele, big R allele, so this enzyme can be used in the starch biosynthesis. All right. However, if something happened here, all right, if there's a mutation, and if this mutation will completely destroy the function of this allele, so of course this is a mutant allele, right? Because little r, and this allele no longer can no longer produce any functional enzyme. So this biochemical pathway will be blocked. Right. So this is one form of the mutation. So just what probably maybe because of the radiation. So there's a one single base change, and this single base is so important for this gene. So if there's a change here, the function of this allele of this gene will be totally lost. So this is one way for the mutation. There's another possible way for the mutation is like, so for example, in the genome, in the genome of any organism, there exists something called the mobile transposon. So the mobile transposon, so we're going to learn this in the future in more details. Here, I just want to let you know, so some DNA fragment can move from here to there. So that's why we call this kind of like DNA as the mobile DNA or mobile transposon. So if some mobile DNA jumped from somewhere in the genome into this gene, right into this gene, and this insertion can totally destroy or change the reading frame of this gene, And this gene again cannot produce any functional enzyme or protein anymore, and this will give you a mutant phenotype. So this is the molecular basis why for a single gene we could have more than one forms. We could have more than one alleles. Yeah, I hope this is clear. Okay, so at the end of this lecture, the lecture 1, so I'm gonna say something about the biochemistry, interesting biochemistry behind big R and the small r. So this is the famous allele we have learned right from Mendel's experiment, the big R and the small r. And we knew, yeah, the big R, the wild type R can produce a functional enzyme here. So this active enzyme, the function of this enzyme is that this enzyme can convert the unbranched starch into the branched starch. We knew the unbranched starch is sucrose, so it's sweet, but this starch is less sweet, right? Okay, now if something occurs in this allele, mutation, for example, and the function of this gene is removed, and then that means like this allele cannot produce any functional enzyme here. So in this case, the pathway for conversion the unbranched starch into the branched starch is blocked. So in the cell of the seeds, there are a lot of unbranched starch, but not the branched starch. So what happened here? Okay, so here, so if the individual, if this pea plant has the mutant allele, little r, so the consequence is what? Is the level of the sucrose in this line is nearly twofold that of the wild type line, and the increase. So in the mutant plant, there's a significant increased sucrose concentration, and you learn chemistry and physics, right? So if the concentration of the sugar is high, so it will help the cells and the seeds to uptake a lot of water, try to reduce the concentration of the sugar, and this will make the seeds bigger, larger, full of water. However, during the seeds maturation, so this water will be lost. And think about that. So initially it has a very large seeds, but because of the loss of the water, the skin of the seeds will be shrinked. So this means like after the seed maturation, you're gonna see the wrinkled shape of the seeds. But this type of seeds contains a lot of good things. The sweet, unbranched starch. So if you prefer the sweeter piece, yeah, just go for this type. All right. So I think, yeah, this is the end of the lecture 1. Let me stop share this one, and we're gonna continue the second.
I have a question, Professor.
Yes, go ahead.
So for the P example you gave, is this a common trend we see in that the recessive alleles typically code for the inactive version of the enzyme, or is this only specific to the wrinkled peas?
I cannot hear you very clearly. Could you say that again? I heard some like background noise. Sorry. Yeah, could you say that again?
Yeah, so for the wrinkled peas case, you mentioned in the last slide of the first lecture, you mentioned how the recessive allele codes for the inactive version of the enzyme. Is this
That's
like
true.
a common trait amongst like recessive alleles in general for?
Yeah,
like
this is a good question. So I don't know if I give you if I can give you a right answer, but at least for pea plants, probably yeah, this is the common thing. Yeah, but for some other plants, probably yeah, they have different genes to control this types of traits. So yeah.
Thank you.
Sure. Thank you. All right. So sorry. So I really I'm really sorry. So I don't have time to answer your questions during the lecture. So so my thoughts is like, so to be more efficient, I'm gonna finish my slides first, and I will leave like maybe 5 to 10 minutes at the end of the lecture, so you can ask me all questions. So in case you forget your question, so please write down your. So that question was really good. Thank you. So please write down your question on your notebook, and when we finish all the slides, so I will allocate some time for you to ask my question, ask questions. Okay, thank you. All right, let's continue. Yeah. Okay. So okay, so in lecture 1, we learned the first Mendelian law, right? The Mendel one. So the Mendel one says, because we just talked about the Mendel one, so I believe everyone will still remember what the Mendel one says. The Mendel one is talking about the segregation, random segregation of 2 alleles, 2 gene members, right? So in Mendel one, we are actually talking about the behaviors or the relationship between 2 homologous chromosomes. They segregate during the meiosis. Okay, so in chapter 2, we're talking about the Mendel 2, the second Mendelian law. So in this law, we are talking about the relationship or the behaviors between the non-homologous chromosomes. All right, let's move on. Okay, so this is this diagram just gives you a simple summary about what is going on in the monohybrid cross based on the Mendel one, right? The random segregation. So we start from a monohybrid F1 parent, and as you can see, we have 2 alleles here, and during the process of meiosis, these 2 alleles is going to segregate in a random fashion, right? So here and here, you see the segregation, right? If we agree that this segregation is a random event, then this can produce 2 types of gametes. So one type of gametes carries the capital R, which is the wild type allele, and the other half will carry the mutant allele, right? So 50-50, right? This is Mendel one. So remember, we are talking about 1 gene, so that's why we use the monohybrid cross, and we can use this beautiful square, the Punnett square, to help us to digest the genetics behind. All right. Okay, now Mendel 2. What's the difference? What's the difference here? All right, here the name is different, right? We do not use the monohybrid, we use the dihybrid. Why is that? Because we're talking about 2 genes, and these 2 genes are located on 2 different chromosomes. So here we still have the shape gene, big R, small r. So these genes are on 1 chromosome, one pair of the homologous chromosome. But we have another gene, so here, so the capital Y and the lowercase y. So this is another gene, and this gene will determine the color of the seeds. All right, so if you look at here, so the R gene will determine the shape of the seeds, but the Y gene will determine the color of the seeds, right? So yesterday, Lei went over, so review the meiosis process with you in the discussion. So every one of us should understand this process, right? So during the meiosis, the homologous chromosome pairs are going to be paired together, and these pairs will be lining up along the cell equator here, okay, cell plate here, right? Okay. However, So if we are talking about two genes on two different chromosomes, so if this cell is gonna go through the meiosis, there are two possible ways for lining up these two chromosome pairs, right? So this is the first possible alignment, right? So in this alignment, the big R is on the same side with the big Y. And the small r is on the same side with the small y. So if the alignment is like this, if this, the option 1 looks like this will gives you two possible gametes here. One is the gamete with the capital R and the capital Y. And the other gamete will give you the lowercase r and the lowercase y, right? Okay, however, there's another possibility that this chromosome pairs can be lining up in this option 2. And in this case, as you can see, the capital R is on the same side with the lowercase y. And the lowercase r and r is on the same side with the capital Y. All right, if the homologous chromosome pairs are lining up in this manner, then it will give you two possible gametes here. So the first gamete is the big R, small y, and the second gamete is the small r, big Y. All right. If we think this process is completely random, which means you have 50% of the chance going this way, you have 50% of the chance to go in this way, and then you can end up with 4 possible outcomes, right? 4 possible gametes here with equal chance. So each type of gametes has 1/4 of the probability. I hope there's no problem for understanding this diagram. All right. Okay, now compared to the monohybrid, so this is dihybrid. We're talking about 2 genes. So it's a little bit more complicated, right? So now to digest this genetics, we cannot use the 2x2 Punnett square anymore. We have to use a more complicated Punnett square. So here we will use a 4x4 table, 4x4 Punnett square to help us to understand what is going on behind this dihybrid cross. Okay. Right. So this is what is happening here. So we still have 2 parents. Let's female parents, lady first, and male parents here. Okay. And so let's go back. So here for each parent, each F1 parent, the genetic makeup is identical, right? So for each parent, there are 4 possible gametes with equal chance. So let's move here. So for the female parents, these parents can produce 4 possible gametes here with equal chance. So then along the y-axis, you got this. And this is the same for the male parents. So the male parents can also produce 4 possible gametes here with equal chance. And then you combine this 4x4, you're going to end up with 16 possible outcomes, genotypes for the progeny, right? For F2 progeny. But if you look at the phenotype, so there are actually only 4 phenotypes here. So the first one is round yellow, the second one is round green, third one is wrinkled yellow, and wrinkled green. And if you check this table, this Punnett score, you're going to see if 1/4 is true for both parents, you are going to end up with an expected ratio of this. So this is the expected ratio. So in the ideal situation, you're going to see 9:3:3:1 ratio. So this is another magic ratio. I hope everyone understand this and remember this, because this is important. All right. So I just mentioned, so if we believe Mendel's 1 and the two laws are making sense. All right, if those two laws are true, then we're gonna see the expected 1/4 probability for each gamete, right? And then if this is true, then we're gonna end up with this phenotypic ratio. But this is the ideal situation, so that's why we call this is the expected ratio. So we're gonna talk about this expected ratio or expectation in statistical analysis. All right. However, when Mendel did the experiment, so this is the real data he observed. So can you see from this data, can you say this data is in agreement with this ratio? Not really, right? So then what should we do? So the next step, we're gonna test this data. If the observed numbers here agree with this expected ratio. So if this data, if this experimental data agree with your expected ratio, this will be the evidence to support your hypothesis. So what are the hypotheses here? So our hypothesis is the two Mendel laws, right? So the Mendel actually proposed the two laws, but he didn't know those two laws are correct or not. So he has to conduct some experiments to collect the data and use data and let data speak, and use data to support, to test whether his hypothesis is true or not. So this is what we're going to learn next. Okay. All right. As I mentioned, this is the expected ratio, expected fraction for the phenotypes, right? And this expected fraction are based on what? Actually, this expected fraction are based on its Mendel laws. So if his laws are true, then we can get this expected ratios or fractions. Okay, now second step, he did the experiment and get these numbers, data. The third step is to use this data to test if this data is supporting this expected ratio or not. If it is, okay, then we successfully prove his hypothesis or the two laws he proposed. All right. So here, here we will learn how we can use this data to test this hypothesis. Usually in science, any project will start from a question. So if you try to answer that question, there are two possible ways to answer that question, yes or no, right? The same here. So we have two answers to the question. The one answer is yes, the data agree with the ratio. In another way says, okay, Mendel's law makes sense. And another possible answer to this question is no, the data do not agree with the ratio. This is equivalent to say Mendel's law doesn't make any sense, right? So we want to test this hypothesis based on his data. So before we analyze his data, so I'm going to show you examples. So this is my favorite example. So by showing this example, we can learn what is going on behind a classical statistical hypothesis testing, right? So the hypothesis testing is actually very similar to what is going on in the court in this country. So we put this gentleman on a trial. We want to see if he is innocent or not. So this is the question we start from, right? So every project will start from a question. So this is the question we try to address. Is he innocent? So for this question, we have two possible answers, yes or no, all right? Okay, and we say these two competing hypotheses. So we name these two competing hypotheses as non-hypothesis versus alternative hypothesis. All right. by doing the hypothesis testing, we do not need to test both hypotheses. We just pick one hypothesis and put this hypothesis on a trial. So this can help us to complete the hypothesis testing. All right, let's say we pick this one as the non-hypothesis. We believe, first, at the beginning, we believe this guy is innocent, right? So this is our non-hypothesis. We start from here. Okay, based on this non-hypothesis, by assuming the non-hypothesis is true, so we collect data. So this is sampling. So if this non-hypothesis is true, if he is innocent, so we're gonna collect the data based on this hypothesis, and then we're gonna test this data by this hypothesis. All right, we're gonna feed this data with the hypothesis to see if your data is supporting your non-hypothesis. So if the p-value is greater than 0.05, which means your data is very supportive of the hypothesis. All right, which means, yeah, this guy is really innocent, so we're gonna set him free. However, if your data does not agree with the non-hypothesis, which means the p-value is less than 0.05, then we're gonna reject the non-hypothesis. What is the non-hypothesis here? So the non-hypothesis is this guy is innocent, right? If your data is not supportive of this hypothesis, p is very small, then you're gonna reject the non-hypothesis and then take the alternative hypothesis and then put him in jail. All right, so here, so actually the p-value, so this is yeah, I think this is the right timing for me to introduce p-value. So the p-value here, I believe many of you have already heard this term. The p-value, so we can understand, we can think about p-value as the probability that how likely your data is supporting your non-hypothesis. So if p-value is large, means like your data is very supportive of your non-hypothesis, then you have enough evidence to accept your non-hypothesis. If the p-value is extremely low like this, which means your data is totally contradicting with your non-hypothesis, which means you have to reject your non-hypothesis based on your collected evidence. All right. So how can we test your data based on the non-hypothesis? This is another interesting example I can show you to help you understand the test. So we tested the hypothesis by feeding the data with the hypothesis or the model based on the hypothesis. Right. Suppose you got an invitation to attend a party tonight, and after work you really do not have time to prepare yourself, but you need to dress you up. So you, after work, you rushed into a department store, and you have to pick one dress from two. So yourself is the data, and you have two dresses here. So you have to select one dress from these two, but you really do not have time to try both dresses. So then you're gonna pick one and put one as the non-hypothesis, and the other one is the alternative hypothesis. So you just try on the non-hypothesis, the first dress. So if you put the dress on and look into the mirror, and then you're gonna see if this dress fits you or not, right? So when you try to fit yourself into the dress, so you're gonna see the difference between you and the dress. If there's, but if the dress is too tight, which means there's little room, very little room between you and the dress, or if there's too much room between you and the dress, means that the difference is too large. This will give you an indication that this dress doesn't fit you. So in this case, you're going to reject the non-hypothesis and just take the alternative dress and run away, go to the party. So if this is the case, the p-value is what? The p-value is like, so when you put on your H0, the non-hypothesis dress on you, So the probability is we can think about the probability p-value as the probability that you will become the focus of the party if you put this dress on. If the p-value is large, which means, oh, you like this dress, and if you just put this dress on, you will become the focus. Otherwise, you don't think, yeah, the dress fits you, so you're gonna throw it away and take the other one and run away to the party. All right. So this is how we tested the hypothesis based on fitting the data. Okay, so let's look at another example to learn how we can do the calculation. So once we have the data, we have the model. So how we can use the statistical test to help us to do the calculation to test the hypothesis. So this is a very, very useful example here. Suppose there's a guy, he likes gambling. One day he wants to test if he has a dice, and one day he wants to figure out whether this dice is a fair one or not. So now this is the question he starts with, right? Is this dice a fair one or not? So there are two possible ways to answer his question. One way is, yes, the dice is a fair one. Of course, the competing hypothesis, another answer, it will be the dice is not a fair one. So now he has two hypotheses and he wants to put the non-hypothesis on a trial. So what he has to do is to do experiment to collect the data. So he's very diligent. He rolled the dice for many times, 600 times. That takes a lot of effort, right? And this is the data he got. So let me give you 3 seconds to think. So based on this data, do you believe this dice is a fair one or not? So let me guess. So I believe most of you would think, yeah, this dice is very likely to be a fair one. Yeah, I agree. So, but why is that? So actually, in your mind, you are actually comparing these numbers with these numbers, right? So what are these numbers here? So these numbers, yeah, these magic numbers actually are the expected number. So what is the expected number? The expected numbers means if your non-hypothesis is true. So what would be the expected number for each of these outcomes? Ideally, right? So in an ideal situation, if this dice is a fair one, you're going to expect to see 100 times showing up for each of the numbers. But in reality, yeah, this is very unlikely to get this number because of the random error. Okay, all right. But if these numbers are close enough to these expected numbers, then you will be okay, right? You can say, okay, there's no problems. So we still believe this dice is a fair one. But if these numbers are too much removed from these expected numbers, then you're not going to be trust the non-hypothesis, right? You trust in the non-hypothesis. All right. Okay, so this is, let me clear this.
Oh, Professor?
Yeah, go ahead.
So then when it comes to the dihybrid cross, for example, the 9-3-3-1 ratio seems pretty close to the results. What kind of deviation are we looking at so that we can draw the conclusion that the dihybrid cross was unsuccessful?
Yes, that's a good question. So I'm gonna address this question right now.
Okay.
Yeah, the deviation. Yeah, because I try to use this example to show you the deviation. Because here we actually, in your mind, you actually compare this number with this, right?
No. Well, yeah. Yeah.
Oh, okay. That's the deviation. So if you take the difference between these two numbers, that's the deviation. So we are going to use this formula to do the calculation chi-square test. So at the beginning, when I throw this equation to you, probably you will be scared. This is too complicated. But once I explain what is going on in this formula, so you're gonna love it. You're gonna appreciate, how beautiful this equation will be, because this is agree with what we are thinking about the problem. So here, so if you look at this equation, so we actually compared observed number versus the expected number by taking the difference between these two numbers. So if this number is very close to this number, if you take the difference, so the difference between these two numbers should be close to 0, right? That's the deviation. If this number is not close to the expected value, then the difference should be very large. Do you agree with that? Yeah, but sometimes, the deviation could be positive or negative. So if you add up all the deviations together, some negative or positive deviation will be canceled out. This will not truly faithfully reflect the data with the model. So that's why we put a square here. So no matter you get a positive deviation or negative deviation, we're going to square this, and everything will be positive. And also we divided the difference, the squared difference, by using the expected value to standardize this quantity. And we will do this calculation for each cell. So we have 6 cells here. So we do this, and for each cell, we calculate this chi-square statistic. Look at here. So we calculate the difference between this two first, and we square it and divide it by the expected value here to normalize this, to standardize this. And we repeatedly do this for 6 categories, add up these numbers together to yield a chi-square statistic. All right. So let me ask you, so if you see a very small number of the chi-square, what do you think? Can anyone tell me? Yeah, if you see a very small number here, means the difference between the observed value and the expected value should be very small, close to 0. And this will give you evidence the data agree with the expected value very well, right? However, if after calculation you get a big number, let's say 600 here, what does that mean? So this 600, the number of 600 will be translated to, okay, the difference between the observed number and the expected value are very large. So in this case, you do not think your data is supporting your null hypothesis, and you're going to reject the null hypothesis and say this test is not a failure, right? Okay, so actually we just calculate this test statistic and use this test statistic, chi-square test statistic as the evidence to show how well your data support your null hypothesis. All right, in the chi-square calculation, you need another quantity here, which is degree of freedom. And this thing is defined by the number of outcomes minus 1. So here we have 6 possible outcomes. The degree of freedom equals to 6 minus 1 equals to 5. All right, so this is another important thing here. So once we got the value here, the chi-square test statistic and the degree of freedom, how can we get a p-value for the test, right? So this is a table we need to use for the calculation. In this case, our degree of freedom equals to 5. So that means in this table, you only need to look at this line. So this line gives you the degree of freedom equals to 5. And the number we calculate here equals to 6.12. And we just find the values on this line, on this row, which are closest to this number. So the 6.12 is in between these two numbers, right? So that means like the p-value associated with your test will be some value in between 0.1 and 0.5. Okay, now the p-value is not less than 0.05, which means the data is quite reasonably supporting your non-hypothesis. So in this case, we do not reject our non-hypothesis. And we believe this dice is a fair one. Okay, I think I'm gonna stop here because we have 5 minutes left. We're doing pretty good. So next time I'm gonna continue this. And so far, do you have any questions so I can help answer?
Yes. Oh, sorry, you go first.
Sorry, I just had a quick question.
Yep.
Referring to the chi-square number, you were talking about it being small or large. Is that similar to standard error?
Yes, kind of, yes.
Okay,
So-
and then just to understand is just that's simply testing for the correlation between, oh no, the distance. Well, yeah, wouldn't it be correlation, right, between the two results?
That's right. So yeah, I would guess you probably have taken some statistics class, right?
Yes.
Yeah. So the standard deviation you are talking about is related to the normal distribution. So we use this term very often in the student t-test, the z-test, or the correlation test. Yes. But here we use a different test. We use chi-square test. So that's why there's no standard deviation in this situation. But we do check the difference between the observed value and the expected value. And then we use this distance in each category to calculate the chi-square statistic. Actually, this value will show how close your observed value is to your expected value. So that means like if you end up with a very small number, which is good, means the data is quite supportive of your non-hypothesis. Otherwise, if you get a big number, which means the difference between here are large, and when you calculate chi-square, the sum, each item of in this equation will contribute a lot to this final statistic, right? Will make this number very large.
Okay. Okay. Thank you.
Sure. Okay. I hope it makes sense.
Yes. Yes, it does. Thank you.
Okay. Thank you.
I just had a. Oh, sorry. Someone else is going.
Oh yeah. So I just want to know what sort of correlation we're making with the number that we get from the key value and how it applies to the chart. Because
okay.
I get that if we have this value 6.12 and it lines up with the degree of freedom of 5, got from the number of what was it, the number of outcomes.
Sure. Okay, so yeah, let me repeat this.
In between 0.1 and 0.5, I just don't know what that means.
Yeah. Okay. So let me explain this a little bit. So so I hope I can make it clear. Otherwise, yeah, next time I will repeat this because this is very important. Definitely, I'm going to put some questions like this in the exam to test you if you understand the statistical test behind the Mendel's rule, right? Okay. So now you understand how we got this value, right? So this is the value we calculated using this formula. So this value actually is a measure, is a ruler. We try to use this ruler to measure the distance between the observed value and the expected value. So this is one important thing you need to know for the chi-square test. Okay, so this is the data evidence. But in order to use this data to calculate the p-value to help you make the conclusion for the test, we need another thing, the degree of freedom. So this is a very complicated concept. So I don't think I have time to make it clear today. But if you are interested in that, next time I'm gonna bring some extra material to describe this. So this degree of freedom is required for the chi-square test to get a p-value. So simply, in each case, how do we figure out the degree of freedom, the value of the degree of freedom? So for example, here, if this experiment will generate 6 possible outcomes, then your degree of freedom will be equal to 6 minus 1 equals to 5. So for example, in the Mendel's experiment, you have 4 outcomes, right? 9 to 3 to 3 to 1. So in that experiment, you have 4 outcomes. So when you do the chi-square statistic for that data, the degree of freedom will be equal to 4 minus 1 equals to 3. Okay, this is the difference. All right, now once you have calculated the chi-square statistic and the degree of freedom, so you can use this table as a tool. So right now, if you are if you know how to do the calculation using computer, so you don't even need to bother using this table. But this is the traditional way we use to help us calculate the p-value based on the chi-square statistic. All right. So in our previous example, the chi-square statistical value is 6.12, and the degree of freedom. The degree of freedom equals to 5, right? So in this case, in this table, this column represents the number of degree of freedom. Since we have degree of freedom equals to 5, so we only need to look at this row. Okay, so on this row, there are many numbers here. So what numbers do we need to use? So you need to find the values that are very close to your value. So which is here, right?
Yes.
Yeah, you do not find the exact value you get, because if you have different data, you definitely will end up with a different value. It's impossible to include all the numbers in this table. I mean, if that is true, you're going to have a large table. But yeah, so we often use this table, simple table for the calculation. So in this situation, you just find the numbers that are closest to your number.
So that, at this point, I think in what range of the p-value are we looking to land in here, right? Because if the chi value was any less, we would have landed closer to 1, correct?
So yeah, this is a good question. So do you see on this row, the values are increasing monotonically, right?
Yes. But
okay.
The p-values decrease as the lower values increase.
Right. That means like if degree of freedom equals to 5, if your chi-square statistic equals to this, exactly equals to this will gives you the p-value of 0.5. If your calculated chi-square statistics is exactly equal to this one, this will gives you the p-value of 0.1. But your value is in between. What does that mean? That means your p-value will fall in between 0.1 and 0.5.
Well, yes, I understand that. It's just what I want to know is what range of p-value are we looking for verification here, right? Because if I'm thinking if it lands closer to 1, then it's more correct, and if it lands farther away from 1, then it's not as correct.
I see.
I'm looking at the p-value 0.995, which is close to 1, and 0.005, which is kind of far from being 1.
That's a good question. So now to answer, before I answer your question, let me describe the definition of p-value again. So in a hypothesis testing, so there's a simple way to understand the p-value. The p-value can be looked as the probability that your non-hypothesis is true based on your data. So based on your data, what is the probability that your non-hypothesis is true? So if the p-value is large, close to 1, which means, the chance, based on your data, the chance that the non-hypothesis is true is pretty high, right? And you're going to accept the non-hypothesis. But if this p-value is very low, which means your data is not supporting your non-hypothesis, which means based on your data, the probability that your non-hypothesis is true is pretty low. Then another question, how low is low? So we need to specify a criteria to help you to make the final conclusion, right? So
Yes.
usually, yeah, the rule of thumb is we use 0.05. I hope like you heard of this criteria before.
No, I haven't.
Oh, okay. So probably you haven't taken any statistics class, right?
No, I'm taking it right now.
Oh, okay.
Yeah.
Good. So in the near future, your statistical professor will talk about the p-value. So 0.05 is a commonly used criteria. We call this a rule of thumb. So back to my second question. So how low is low? Because we need to find a cutoff to make the conclusion. Either you're going to reject the non-hypothesis or you're going to accept the non-hypothesis, right? So,
Yes.
so if your p-value is greater than 0.05, yeah, it's safe for you to just accept the non-hypothesis. Otherwise, if the p-value is lower than 0.05, reject it.
All right. Okay, cool. That actually clears up a lot for this concept.
Okay, great. So let me see, what time is it now? Oh, wow. I'm sorry. So, okay, so I think, yeah, we're doing pretty good today, and but still, like, I couldn't finish all the stuff. But yeah, next time, let's continue talking about the chi-square statistical test, and then we're gonna show how we use this tool to test Mendel's two laws. All right, thank you so much. Let me see. All right, see you next time. Bye-bye.